So let's start the chapter, the cell, the unit of life. So this chapter is a continuation of what you have learned in 8th and 9th standard, that's cell. And in this chapter you will be studying about both prokaryotic and eukaryotic cell structure. So what is the main difference between a prokaryotic cell and a eukaryotic cell? The main difference or the main basis of classification of prokaryotes and eukaryotes is the structure of nucleus. If the nucleus is not bound by a nuclear membrane, if there is no true formation of chromosomes, or if the membrane-bound cell organelles are absent, then such group of cells are called prokaryotic cell and the organisms are called as a prokaryotes. Now, under the group of prokaryotes, there are four main groups, that is bacteria, cyanobacteria, mycoplasma and PPLO, that is pleuropneumonia-like organism. Actually, this mycoplasma and PPLO both are almost the same only. They all have the common character that their nucleus is not well defined. Okay. Now, to study the prokaryotic cell, we are taking a typical example of bacterial cell. Now, bacteria based on their shape, there are four different types. Round shaped ones are called as coccus. Then rod shaped ones are called bacillus. Comma shaped ones are called as vibrio. And spiral shaped ones are called as spirillum. Now here we are taking the example of a rod shaped bacteria, that's a bacillus type of bacteria, for studying the cell structure of a prokaryotic cell. A very common example for a bacillus type of bacteria is E. coli, otherwise called as Escherichia coli. There are different strains of this bacteria. A few types of bacteria are present in the human intestine, which are actually beneficial for human beings. There are other strains which can cause diseases also. So anyway, so this is a typical bacterial cell structure. Typical means നമ്മൾ ഒരുപാട് പേരെ കൺസിഡർ ചെയ്തില്ല, അല്ലെങ്കിൽ മെജോറിറ്റിക്ക് ഉള്ള ഒരു characteristic feature ഉള്ള ഒന്നിനെയാണ് we call it as a typical. So we can explain this or take this as a typical cell structure of a prokaryotic cell. Now, what are the parts of a prokaryotic cell? So these are the main parts coming under the prokaryotic cell. It will have a cell envelope. Then there are structures called as lysosomes or chondrioids. Then ribosomes are present both in prokaryotes and eukaryotes. Then inclusion bodies are there. Genetic material, about it we will study in detail. Then flagella, pili, otherwise pili also we can pronounce, and fimbriae. So these are the parts present inside a typical bacterial cell. Now we will learn one by one. First of all, the cell envelope. The bacterial cell envelope is having three important layers. The outer layer is called as glycocalyx, which can be either a slimy layer or it can be capsule. രണ്ട് ടൈപ്പിൽ കാണും. ചിലതിൽ we call it as a slimy layer. The glycocalyx can be slimy layer. എന്ന് പറഞ്ഞാൽ അതിനൊരു definite shape ഇല്ലാതെ ഒരുമാതിരി diffuse and slimy ആയിട്ടിരിക്കും. So it's loosely fixed to the bacterial cell. അങ്ങനെയാണെങ്കിൽ നമ്മൾ slimy layer എന്ന് പറയും. Capsule എന്ന് പറയുന്നത് when it is rigid and fixed tightly to the bacterial cell, we call it as capsule. So glycocalyx will be there. It can be as a slimy layer or as a capsule. Then comes the cell wall. Cell wall is made up of, it's not like plant cell wall like cellulosic cell wall alla ivide fungus ulladu chitin cell wall aanu ivide adu alla. We have this bacteria is having a cell wall made up of a chemical substance called as peptidoglycan. Now along with the peptidoglycan there is N-acetylmuramic acid and N-acetylglucosamine. So these are the components that makes up a bacterial cell. And this peptidoglycan cell wall gives that fixed shape and rigid nature for the bacterial cell. And it is also porous. Porous means some type of substances can be moved in and out through the cell wall. That's why it is called as a porous cell wall. Now, many of the bacterial properties are associated with the cell wall. Means if cell wall is not present in a bacterial cell, bacteria cannot survive. So cell wall is very important for a bacteria. That's why many antibiotics like penicillin and all interrupt the cell wall formation in bacteria and thus kill the bacteria. Okay, so that is all about the cell wall. Then comes the third inner layer, that is the cell membrane. So as in the case of eukaryotic cell, the cell membrane is made up of phospholipids and proteins. It is selectively permeable membrane, means it allows only certain materials to pass in and out. It is a living membrane, and all these are the characters of cell membrane. Now, in bacteria, which is a prokaryote, many membrane-bound structures or almost all membrane-bound structures or organelles are absent. So, the cell membrane forms infoldings called as mesosomes. It is also called as chondrioids. So, mesosomes are otherwise called as chondrioids. So, what are mesosomes? They are the infoldings of cell membrane. Then they are of two types, septal and lateral. Septal mesosomes are, this is the mesosome. So you are seeing this layer, it is the glycocalyx layer. Then the next line shown is the cell wall, and this third line shown is the cell membrane. So the cell membrane form infolding called as the mesosome. Now this mesosome is of two types. It can be septal or it can be lateral. That is, if the mesosome grows down touching almost the center of the cell in the genetic material region, then we call it as a septal. The function of septal mesosome is to help cell wall formation. It also helps DNA replication. What's the meaning of DNA replication? A DNA making its own copy is called as DNA replication. When a cell divides, the genetic material should replicate. So that newly formed cell also will get a copy of it. So this DNA replication is helped by the septal mesosome plus the distribution of DNA to the newly formed cell equally. That is also done by the septal mesosome. Then there is another mesosome called as lateral mesosome. What is the function of lateral mesosome? Since mitochondria is absent here, this lateral mesosome contains certain enzymes which are important for respiration in bacteria. So that is about the mesosomes. Now coming to ribosomes. What are ribosomes? While ribosomes are actually particle-like structures, it is not a membrane-bound structure. So it is present in both eukaryotes and prokaryotes. In case of eukaryotes, like a plant cell, it is present free in the cytoplasm. It is seen attached to the endoplasmic reticulum. It is seen inside the mitochondria, and in a plant cell, it is seen inside the chloroplast also. But in case of prokaryotic cell, that is a bacterial cell, ribosomes are seen free floating in the cytosol. Cytosol is nothing but the cytoplasm. So a cytoplasm free to float here, we can see the ribosomes. Now ribosomes in their natural condition, they exist as two subunits. Always they are like that. There is a small subunit and a large subunit. At the time of ribosome, the main function of ribosome is protein synthesis. So protein synthesis time over, the two subunits will come together and help the process of protein synthesis. In prokaryotes ribosome, it is of two, that is the smaller subunit and the larger subunit is there. The smaller subunit is 30S type and the larger one is 50S type. They join together and make a 70S type. The S in one is Friedberg. Friedberg is the unit, or we can say sedimentation coefficient in the unit. Don't think that when it is added it is not 70, then how it is. It's not like that. There is sedimentation coefficient value on a parameter. So that is different from that present in the eukaryotic eukaryotes. Ribosomes in eukaryotes are slightly larger than those present in the prokaryotes. So wherever ribosome is present, it is concerned with protein synthesis. Now, in case of this bacteria, we can see that ribosomes are sometimes seen as a group. That is, two or more ribosomes are seen attached to an mRNA. What is mRNA and all? You will study in 12th standard in detail. mRNA means messenger RNA. Messenger RNA is actually containing the information that is needed for protein synthesis. So mRNA is produced from a DNA molecule, and this mRNA is having the information for the synthesis of a protein. So same way here also, the mRNA usually in case of eukaryotes, mRNA will be attached to a ribosome, a single ribosome. But here we can see that a group of ribosome is attached to the same mRNA. And the same protein will be synthesized simultaneously by all these ribosomes. So of ribosomes attached to the mRNA is called as polyribosomes or polysomes എന്ന് പറയും. Okay, so their function is also protein synthesis. Then comes the next thing, that is inclusion bodies. What are inclusion bodies? Inclusion bodies are usually non-membrane bound structures. It's not covered by any membrane. They are actually storage structures in a bacterial cell. They may store glycogen or phosphate granules will be there, or cyanophysin granules will be there in the group of cyanophysae. So like that they store something. Then in some aquatic forms of photosynthetic bacteria like green and purple bacteria, you can see that gas vacuoles are also formed in them. They are also considered or put under this inclusion bodies in some textbooks, and they help the bacterial cells to float on surface of water. They are seen, gas vacuoles are seen only in aquatic forms of bacteria. Then comes the main thing, that is the genetic material. So genetic material, unlike in the case of eukaryotes, here there is a single molecule of DNA which is loosely arranged into loops held by certain proteins in the central region of the cell. But there is no covering for it, a definite nucleus is not formed. This region is called as nucleoid. So what is nucleoid? Nucleoid is a region in the bacterial cell where you can find the single circular DNA arranged as big loops held by certain proteins. So this region is called as nucleoid. There you are seeing the single DNA molecule. In case of eukaryotes, that also you will study next year, the chromosome is formed, DNA forms chromosome with the help of certain proteins called as histone proteins and non-histone proteins. So here such structures are not there. So we call the DNA in prokaryotes as naked DNA because there is no histone molecules in that. Histones in the proteins are. So histone proteins are not associated with the DNA in case of prokaryotes. So we can see a single DNA, or sometimes we use the term single chromosome also. But the actual chromosome formation is not taking place here. Other than this, there are extra chromosomal, small circular double-stranded DNA like this. These are called as plasmids. It can be one or many in a cell, and these plasmids contain certain genes also, which are very essential for the survival of the bacteria. For example, some plasmid contain some antibiotic resistant genes. So when bacteria infect our body, we use antibiotics. So antibiotic resistant genes ulla plasmids can resist the antibiotics. Okay. So these are called as plasmids. In the speciality, it can replicate its own, not considering the replication of the cell or the DNA inside the main DNA. It can replicate its own and make as many number of copies as it wants. And it can also transfer this plasmid to other bacteria by some methods. So that is about the plasmid and the genetic material. Then next thing we have to study here is flagella. So flagella can be one or many in a bacteria. They are actually structures which help locomotion in bacteria. A flagella is having mainly 3 parts are there. This flagella is having the main fiber-like part. Then there is a hook-like region, and then there is a basal body. So 3 parts are there. Then this part of flagella is seen on the surface of the cell. Then there is a portion in the base portion of this flagella is called as the hook, and hook is attached to the basal body. This is the basal body which is seen inside the cell envelope. So its function is to help the bacteria move. Then other than this flagella, there are some other structures called as pili also is right pronunciation, and fimbriae. Both are same only. So these are small hair-like structures seen all around the body of bacteria. Its main function is to attach to some specific surfaces, maybe to some other bacteria or to the host cell and all. So they are mainly concerned with attaching to some other surfaces or to the host cell or to some other bacteria. Some pili or pili mainly, they are called sex pilis. So they help to bring two bacteria close to each other for the purpose of conjugation, that is transferring genetic material from one bacteria to another. So for that also pili or fimbriae is used. So these are the parts coming in a typical prokaryotic cell. Based on the staining property, there are two types of bacteria. There is gram-positive bacteria and gram-negative bacteria. There is a person called as Hans Christian Gram. So he is the person who developed the staining process in bacteria. So he developed the stain, and our stain, when we stain that, some bacteria will get a purple stain, the other will get a pink stain. So those bacteria which get stained purple are called as gram-positive bacteria. The gram-negative bacteria usually get a pink color during the staining process. The difference is that the gram-positive bacteria is having a thick layer of peptidoglycan and there is no lipid covering above it. Whereas in case of gram-negative bacteria, there is a very thin peptidoglycan layer and there is a covering of lipid over that cell wall layer. So that's the specialty of gram-positive and gram-negative bacteria. So these are the main things coming under the typical cell structure of a prokaryotic cell.